we are going to go ahead and get started. So for today's lab, we are going to be taking a look at genetics. So I'm gonna go ahead and take over here. So hopefully you guys can see the PowerPoint now. Okay. Is everyone able to see the PowerPoint? Yeah, okay, cool. All right, so so for today's lab, we're going to be taking a look at genetics. And so genetics is a topic that we're not going to be able to get to until after spring break. So we're going to kind of get introduced to it a little bit today and kind of look at the different aspects of it. And then we're going to look at it a lot more in detail when we come back from spring break in 2 weeks. Okay, so this does get a little bit tricky and a little bit complicated. So we're going to kind of take our time with this, and we're going to go through a few different practice problems with this. So if you guys have any questions, feel free to stop me, and then hopefully I can kind of make this make sense here. Okay. So genetics. So genetics is going to be the study of how different traits are going to be passed down from generation to generation. Okay, from parents to their offspring, and then from their offspring to their own offspring, and so on and so forth. Okay. So I'm going to kind of skim through the introduction here, and we'll talk about the introductory portions of genetics more in lecture. But I kind of want to get through this so that we can kind of get a little bit more with the practice. But essentially, there was this guy in, I believe, the late 1800s. His name was Gregor Mendel. And so he is considered the father of genetics. He's the one who kind of really studied genetics in its, in a large, kind of to a large scale. And his work kind of set everything up for how we're able to study genetics today. Okay. And so the way this kind of works here is what he was able to do is he was able to find that there were homologous chromosomes in our cells, right? So remember, when we're looking at homologous chromosomes, we are going to have two sets of chromosomes. Okay, so I'll go ahead and draw one over here. Ooh, let's try that one again. So we should have two sets of chromosomes. So here's one, and then a second set of chromosomes, kind of looking like that. Okay. And so this would be one pair of homologous chromosomes. Okay. So for each of these homologous chromosomes, okay, one came from mom, so I'll just put M over here, and then one of these came from dad. Okay. So when we're looking at our cells are going to have homologous chromosomes in them. Okay. And so these chromosomes will be inherited from our parents. Okay, one from mom and then one from dad. Okay. Now, when we're looking at all of our chromosomes here, our chromosomes are going to have like several hundreds of genes. Okay, sometimes even thousands of genes. And each of these genes is going to code for a specific characteristic in a particular organism. Okay. So remember, in a homologous chromosome, we're going to find the same genes in the same locations. Okay. So let's just say this is where we'd have a gene on mom, and then a particular gene on dad. Okay, same in the same genes, same locations, right? Now, again, when we're looking at our chromosomes, our genes are going to be contributing to specific traits. But as we know, even though we have the same genes, our genes are going to be a little bit different. We're going to have different variations of that gene. Okay. Those variations of these genes are what we refer to as alleles. Okay. And so these alleles are going to be variations of a particular gene. So for example, if we're looking at our example here on the right, we're going to have a gene for flower color. And so every plant is going to have a gene for flower color. Okay. But there's going to be a specific gene, okay, or a specific allele that's going to be purple. So that's going to give you the purple flower allele. And then here we have some white flowers. So we're going to have a white flower allele. Okay. So again, they're all coding for the same trait, okay, the same characteristic, but a different variation of that characteristic. Okay, so we got a purple allele and then a white allele. Okay. And these are going to be found on homologous chromosomes. So when we're looking at these alleles, we can classify them in one of two ways. Okay. An allele can either be considered dominant, in which case we would signify it with a capital letter, like we see over here. Okay. Or if it's recessive, okay, recessive meaning that it's would be indicated with a lowercase letter. Okay. Now, when we're looking at our alleles, like I said, these alleles are going to be inherited from your parents because they're coming from homologous chromosomes from each parent. So for that reason, our alleles may either be the same, or these alleles that we inherit may be different. Okay, so for example, if one of our parents has, brown hair and another parent has blonde hair, right, we can end up with having alleles, or two different alleles in our own body cells. Okay, one allele that came from mom, the brown hair allele, and one allele that came from dad, the blonde hair allele. Okay? So sometimes the alleles that we inherit may be different because we're just inheriting different variations of that allele. Okay, in some cases they may be the same. So if we have two parents that both have brown hair, we're going to inherit both brown hair alleles. So we're going to have the same allele in our homologous chromosomes. Okay? Although it is possible to have different alleles. Okay? We have different versions of it. Now, in a population, it's kind of important to remember that in a population as a whole, there's more than just two alleles in a population. Okay, so example, if you're thinking of hair color again, we're going to have people who have black hair, people who have brown hair, people who have blonde hair, people who have red hair. So those would all be different alleles that exist in a population. Okay. So everything kind of making sense so far. You kind of follow me. Cool. Okay. So we just have different alleles, and each allele is going to code for a specific trait, okay, or a specific variation of that trait. Okay, so let me go ahead and kind of clear this out here. Okay, so when we're looking at alleles, our alleles are going to interact with one another, okay, to help contribute to our organism's physical and genetic characteristics. Okay? So when those alleles interact, they're going to give us two things. The first thing that they're going to give us is the phenotype. Okay? The phenotype is the observable trait that we're looking at, that gets expressed by an organism, something that we can actually see. Okay? The second thing that the alleles are going to contribute to giving us is the genetic makeup of the organism, which is called the genotype. Okay? So the genotype is the genetic makeup. That's the actual, parts of the DNA, the actual DNA nucleotides that will contribute to our physical appearance or our physical trait. And so when we're looking at the phenotype and the genotype, what we're going to see is that the genotype is going to determine our phenotype, okay? Which makes sense. Our genetic makeup is going to determine what we look like, our physical appearance. Okay? So we'll give a couple of examples here. So the first one here is the phenotype. So like I said, the phenotype is going to be the physical characteristics that are actually getting expressed by that organism. Okay, so over here, we've got a couple of pea seeds. So the seed color, we have one that's yellow and one that's green. Okay, so we have yellow seeds and green seeds. This would be the phenotype, the actual color of the seeds that we're actually physically able to see. Okay. The genotype is a little bit more complicated. Okay, so the genotype is going to be determined by the combination of both alleles that you inherit. Okay, both one from mom and then one from dad. Okay. So the combination of the alleles that you get is going to be your genotype. So in this case here, we have one dominant Y allele and then one dominant Y allele. And that gives us the genotype, which is two big Y alleles, or a big Y, big Y. Okay. Over here, we have one little Y allele and then a second little Y allele. So that would be our genotype there, little y, little y. Okay. Here we have a big Y allele and then a little Y allele. Okay. So we have one of each, and that's going to form our genotype, kind of big Y, little y. Okay. So we're going to classify all of our genotypes based off of the types of alleles that they're inheriting. Okay? So if we inherit two of the same types of alleles, okay, they're going to be classified as homozygous. Okay? Homo meaning same, zygous meaning kind of DNA. So same alleles, okay, two big Ys. So this would be homozygous. Okay? This one would also be homozygous. Okay, little y, two copies of the same allele. That would be considered homozygous. Okay? Now each of these, even though they're both homozygous, are going to be contributing to forming different traits. Okay? So this first one, this homozygous with two dominant alleles is going to give us the yellow color, and then two recessive alleles, the genotype with two recessive alleles will give us the green color here. Okay? So those are both homozygous alleles, but contribute to different traits, okay, or different variations of that trait. Okay? The last type of genotype that we can have is called heterozygous. Okay? And so heterozygous is where you have one of each type of allele, okay? One dominant allele and one recessive allele. Okay? So that would be considered heterozygous, hetero meaning different. Okay? So if we look at the F1 generation, so we took our parents, so these are our P generation, our parent generation, okay? We allow them to mate, and then they form some offspring. Okay? all of the offspring that we form here are genotypically different. Okay? They have a different genotype than the parents. The big Y, little y. These guys are heterozygous, so they're going to have big Y, little y here. Okay? So they're going to be genotypically different here. Okay? However, we see that all of these individuals in this F1 generation are all yellow. Okay? All of them are 100% yellow, and that's what we're going to end up seeing. Okay? If we allow the F1 generation to just mate with one another again, okay, we take some of these heterozygous individuals, allow them to mate again, then we start to see the green traits start to pop up again. Okay, so we saw it in the parent generation. It skipped the F1 generation and then reappeared here in the F2 generation. Okay, so that's going to be a trait that we're going to be able to see when we look at recessive traits, and we're going to look at this in a little bit more detail here. Okay, however, we do still see a lot of the yellow individuals here, yellow seeds. Okay, so now we're going to classify these a little bit more specifically here. So when we look at these pea plants, okay, the plants in our parent generation, okay, these are both going to be homozygous. Okay, so again, we see big Y allele, and then little y allele, little y allele. Okay, so homozygous means you have two copies of the same allele. Okay, and again, each of these alleles is going to be found on a different homologous chromosome. So one of these is going to come from mom, and then the other one's going to come from dad. Okay, and same with this side over here. And so these are the parents. This is one parent here. This is one parent over here. Okay. And so if we have two copies of a homozygous dominant alleles, okay, two copies of homozygous dominant alleles, we classify that genotype as being homozygous dominant, okay, because we have two copies of this dominant allele. Okay. Now, if we have two alleles that are recessive, okay, two recessive alleles here, okay, then we have what's called homozygous recessive, okay, meaning that we have two copies of the recessive allele. Okay. So that's going to be the way we're classifying it there. Okay. Now, when we allow these two individuals to mate, we have one parent over here, another parent over here. We allow these guys to mate. Okay. Then they end up forming offspring in the F1 generation that have one copy of each allele. Okay, this one was able to pass down its big Y allele. This parent was able to pass down its little y allele. Okay. And so because now we have two different alleles, okay, in this one individual, two different copies, these are what we're going to refer to as heterozygous individuals, okay, for this trait. Okay. Again, you have one copy of the dominant allele and one copy of the recessive allele. Okay. Now, when we're looking at these when we look at these individuals, we find that all of these take on this yellow trait, okay, this yellow allele here. Okay. Even though one parent was yellow and the other one was green. All the offspring come out yellow. Okay. And so we'll talk about that in a second. But before we get there, is there any questions on anything we've covered so far in terms of homozygous and heterozygous and what that means? And all of these alleles and genotypes. There's a lot of vocabulary words that are all over the place here. Any questions so far? No. Okay. So if there's no questions, we'll kind of move forward here. So like we said, we have these heterozygous individuals that have one copy of the yellow allele and one copy of the green allele. Okay. However, all of these individuals are yellow. Okay. Does anyone have an idea why this would be the case? Why are all these individuals yellow even though they do carry the green allele as well? Anyone have an idea? So Kimberly says because the Y is dominant. Yes. So it's because the Y allele is dominant. So we'll talk about why that is in a second. So dominant alleles are going to be able to mask the recessive alleles. So just hang with me here as we kind of explain this. Okay. So when we're looking at homozygous dominant individuals, okay, the individuals who have two copies of the dominant allele, okay, as we see over here, okay, and then the heterozygous individuals, okay, they're going to look identical for that particular trait. So for example, the homozygous dominant alleles or homozygous dominant individuals are both going to be yellow, while all the heterozygous individuals are also going to be yellow. So they're going to be identical here. It's going to be a little hard to tell, just by looking at the two, whether you're looking at a homozygous dominant or whether you're looking at a heterozygous individual. Okay, they're going to look identical. Okay. When we look at a recessive allele or a recessive trait, the only way we're going to see a recessive trait is if we have two copies of the recessive allele being present. Okay, So we have to have a homozygous recessive individual. Okay. The reason for that is when we look at a heterozygote. So a heterozygote, like I said, is going to have one copy of the dominant allele and one copy of the recessive allele. Okay. So in a heterozygote, the dominant allele will always mask the recessive allele. Okay. Always will mask the recessive allele. Okay. And so for that reason, you'll never see the recessive allele if that dominant allele is also present. Okay. The dominant allele will mask that. So that's what we call the law of dominance. Okay. And so that's kind of what we're seeing over here. So here we see, one parent passing on the dominant big Y allele, the yellow allele. The other one's passing on the recessive green allele. And so we have one copy of the dominant, one copy of the recessive allele in the heterozygote. Okay. But because we have a dominant allele, the yellow allele, it's going to be masking the effects of the recessive one. So you won't see any of that green allele present, okay, or that green trait present in this generation. Okay. And same thing is going to be the case over here. Okay, we take these purple flowers and we mate them with these white flowers. Okay, allow them to mate. They're going to form some offspring in the F1 generation, and we're going to see that all of the offspring here are purple. The reason for that is because the purple flowers had to be the dominant allele here, okay, the dominant trait. Okay. So again, the only way that you're going to see a recessive trait show up is if you have two copies of that recessive allele. Okay. If you have one copy, it's going to end up getting masked by the dominant allele. Okay. This kind of makes sense so far. Okay. All right. So that is going to bring us to recessive conditions. Okay. So like we mentioned, heterozygotes will have a copy of the dominant allele and one copy of the recessive allele. Okay. However, even though they still have the recessive allele, they're still able to pass on that recessive allele to their offspring, even though they're not expressing it. Okay. So an example of that is with albinism. Okay. So albinism, or being albino, is a recessive condition. Okay. It's an inherited recessive condition. Okay. Again, the only way that you're going to end up seeing a recessive condition is if you inherit both copies of the recessive allele. So I'll kind of just write little a, little a. Okay. So you have little a, that's how you're going to end up getting albinism. Okay. So I'll just put albi. Okay. So that's how you're going to end up with albinism. Okay. Two copies of the recessive allele. Okay. However, okay, so he has two copies. So this individual, this child over here, has two copies of the recessive allele. He's going to have albinism, so he's not going to have any skin pigmentation. However, we see his mom over here, and his mom has normal skin pigmentation. Okay, so how is this baby able to inherit? Yeah, I know this writing tool sucks. So we have this this parent, this mom over here, and she expresses normal skin pigmentation, but somehow her baby was, or her son was able to inherit albinism. Okay, so what had to have happened is mom had to have been carrying one copy of the recessive allele and then pass that on to her offspring, or off to her baby, and the father also had to pass on another recessive allele, okay, to her baby as well. Okay. Now they have normal skin pigmentation, and so the normal skin pigmentation is going to be the dominant trait. Okay. And so we're going to assume that mom is big A, little a, and dad is big A, little a. Okay, so they're both heterozygotes. Okay. Now they just happen to both pass on their recessive allele to their offspring. And as a result, the baby ended up being albinism, even though neither parent had albinism directly. Neither parent was albino. Okay. So the key is that in order to get a recessive trait, you have to inherit both recessive alleles from both parents. Okay. Everyone kind of following me so far? All right. Okay, so any questions before we go forward here? Okay, so, oops. Okay, so that's going to lead us into this tool that's going to help us predict the different genotypes and phenotypes that our offspring can develop. Okay? And so geneticists are individuals or scientists who want to be able to determine or predict what an offspring's genotype is going to be and phenotype based off of the genotypes of the parents. Okay, they want to be able to determine the likelihood of a baby or a child inheriting, different types of traits. Okay, whether they're going to inherit a specific condition, a disease, a genetic condition, hair color, eye color, various other kinds of different things, right? And so we're going to be able to determine the likelihood or the chances of developing or inheriting a particular trait just by looking at the genotypes of the parents. Okay? So this is going to be using some Punnett squares here. So we're going to take a quick look here at how a Punnett square is going to be organized. Okay? And so when we're looking at a Punnett square, we're going to orient our square with 4 boxes. Okay? So I'll kind of draw out what this kind of would look like here. Well, I guess it's already here, so I'll just kind of worry about that. So we have our 4-box Punnett square. You're going to draw a big box with 4 little boxes within it. Okay, and so along the sides, you're going to have the genotypes for each parent. Okay, so for example, this would be the genotype for dad. So I'll go ahead and just write. All right, dad over here. And then over here, you would find the genotype for mom, and I'll do this in a different color as well. Oops. Okay. So here you have dad's genotype, and then here you have mom's genotype. Okay. Okay. So those are the genotypes. Okay. In each of the genotypes, you're going to have the alleles. Okay. So each of these would be an allele that would be found in one of the gametes. So for example, dad's sperm cells. Okay. Let me put this in blue actually. So this sperm cell that dad is going to be producing would have an A allele in there. Okay. This would be another sperm cell that dad might be producing. Okay. That's going to have a big A allele in there. Okay. And then these would be egg cells that mom would be producing. Okay. So these are the eggs. Okay. So each of these alleles represents what allele would be found in each of those gametes, in either the sperm or the egg. Okay. So then the last thing that we're going to do here is we're going to look at all the different scenarios that can take place, okay, when these sperm and egg meet up. Okay. So and when fertilization takes place. Okay. So what we're going to do is we're just going to cross each of these individual boxes over here. So here we have a big A, big A, big A and a little a over here. So when this sperm cell and this egg cell join up, they're going to have a big A, little a individual. So a heterozygous individual over here. Same thing with this one. So big A, little a, and so this is going to be the resulting zygote after fertilization happens. Okay. Same thing with this box. So big A and then this little a over here. That's going to give us this box. And then big A and little a, that's going to give us our 4th box over here. Okay, so these are going to give us the genotypes of the potential offspring. Okay. And so what we can do after we determine the possible genotypes is we can determine the likelihood of developing a particular genotype and then the likelihood of developing or inheriting that particular phenotype. Okay, and what the chances of developing that phenotype are. So that's how we're going to organize our Punnett square. Okay. Any questions on how we organize the Punnett square here? No. Okay. Cool. All right. So we're going to do a couple of examples here. So here we have situation. So we have in human populations, brown eyes are a dominant trait while blue eyes are recessive. Okay, so Tony Stark and Pepper Potts are having a baby. Tony is homozygous for brown eyes, which means he's big B. Okay. Pepper also has brown eyes, but she is heterozygous. Okay, so what is Pepper's genotype for eye color? Is she big B, little b, or little b? What do we think? If she's heterozygous. Right, so everyone's getting this one. So This would be big B, little b, right? So she's heterozygous, so she's going to have one copy of the dominant allele and one copy of the recessive allele. Perfect. Okay, so same situation. And so we're going to assume, or we're going to try to identify the genotypes of her possible, or their possible baby. Okay, so what we're going to do here is we're going to draw our Punnett square. And I'm going to see if I can do this effectively on here. So here we have our Punnett square. Oh dear, let's try that again. Okay, so there's our Punnett square. So the first thing that we're going to do is we're going to find the genotypes of both parents. Okay, so we know Tony's is homozygous for brown eye, so he's big B, big B. So we're going to write him across the top. It doesn't matter where you put him; it's just preference for this case. Okay. So big B, big B. So this is Tony's genotype. If we're looking for Pepper's genotype, okay, Pepper's genotype should we classify her as heterozygous, and so as we saw, she's going to be big B, little b. Let me do this in another color. So big B, little b. Okay. So now that we know their genotypes, the next thing that we're going to do is we're going to cross all of these guys and see what the possible genotypes we can end up with for their baby. Okay. So I'm gonna go ahead and do this in a third color here. So we'll do this in green. Okay. So we're gonna take this box and this box over here. So these first two boxes would give us what genotype? What would we get in this first box here?
Two big B's.
Okay. About these third box, what genotype would we get in here? Right, we'd be getting big B, little b. Big B, little b. And then same thing in this box over here as we cross big B with this little b over here. Okay, big B, little b. Okay. So now that we've done our Punnett square, okay, now we can determine the types of genotypes that we can get. Okay. And so this is one thing that it's going to be important here. So first of all, let's start with the different genotypes that we can come up with. What's the likelihood, based off of our Punnett square, that we're going to end up with an offspring that is homozygous dominant? What's the likelihood? 50%, right. So it's going to be 50% chance of having a homozygous dominant offspring. Right. Cool. Now, what's the likelihood of us getting an individual who is is heterozygous? So I'm seeing 50%, right? So a 50% chance of a heterozygous offspring. So big B, little b. Okay. What's the chances of having a homozygous recessive individual or offspring here? 0, right? Because we don't see that anywhere in the Punnett square. Homozygous recessive, which is little b, little b. Okay. So that's one thing that you need to be comfortable with analyzing when you look at your Punnett squares, and the different genetic ratios that you're going to come up with. Okay, 50%, 50%, and 0% in this case. Okay. Now let's kind of take this a little bit further. Okay. The next thing that we want to now determine is we want to try to identify what's the actual offspring going to end up looking like. Okay, what's the phenotype going to be? Okay, so looking at our Punnett square, what's the chances that our offspring are going to have brown eyes? So I'm seeing 100%. And so 100% would be correct. Okay. 100% chance of a baby with brown eyes. Okay. And so how do we know it's going to be 100%? Anyone have an idea? Right, so Kimberly got it exactly right. So they all have the dominant allele. Okay, they all have that dominant big B allele. Okay, all it takes is one copy of the dominant allele to give you the dominant trait. Okay, all 4 of these guys are going to have the dominant allele. So as a result, you're going to have a 100% chance of having an offspring with brown eyes. Okay, and as a result, you're going to have a 0% chance of a baby with blue eyes. Okay. Does this make sense so far? Any questions before we go forward here? Okay. All right. Yeah, cool. Okay, cool. All right, so I'm gonna go ahead and clear this out then. We'll go ahead and jump to the next one here. So yes, this is exactly what we would see. Okay, so we see big B, big B, little b, and you would see your Punnett square, all of them having the big B allele. Perfect. Okay, so at this point, all we've done is we've looked at simple dominance and simple recessiveness. Okay. This is only one type of inheritance pattern. So there's actually a lot more, and this actually gets more complicated, okay, when we're looking at different types of inheritance patterns. So we're going to see incomplete dominance. We're going to see codominance. In lecture, we're going to look at multiple alleles in a couple weeks, and then we're going to look at sex-linked traits as well. Okay. So we're going to take a look at all or 3 of these 4 different types. And so the first one is going to be incomplete dominance. Okay. So when we were looking at simple dominance and simple recessiveness, everything we just looked at previously, we're only inheriting one of 2 traits. Okay. And so that was what Mendel kind of concluded, is that when you're looking at inheritance, offspring are always going to inherit one of those individual traits from one of their parents. It's not going to inherit both. It's not going to be a combination. Okay. That's what he concluded, but he wasn't fully correct. He was correct in some senses, in some kind of, in some situations, okay, but in other situations, you actually can have a blend of the 2 alleles. Okay, and so this is what we call incomplete dominance. Okay, so in incomplete dominance, when we have a heterozygote, okay, the heterozygote is actually going to have a phenotype that is going to be a blend of the 2 parents. Okay, so for example, here we have a red snapdragon plant. Here we have a white snapdragon plant. Okay, when they mate together, they're going to form some pink snapdragon plants. Okay, so we actually have a combination. We have a blend of the 2 parents here. Okay, and so this is going to be a situation where one of the alleles is going to appear, okay, but it's not going to exclude the other allele from showing up as well. Okay, both of them are going to be working together to create the phenotype that we see in that offspring. Okay, so I'm going to show you an example here. And so in incomplete dominance, we do not have any recessiveness. Okay, we just have homozygotes and we have heterozygotes. There's no dominance, there's no recessiveness. Okay, and so what this looks like, so we were looking at a red snapdragon plant, it would have the genotype CR. Okay, C just being color, R being red in this case. Okay, so this would be the CR would be one allele, the other CR would be the other allele. Okay, and so we're going to put these along one side of the Punnett square. Okay. The white snapdragon plants are going to be classified as CW. That's going to be their genotype. And so we're going to list that over here along the top part of the Punnett square. Okay. So we have red snapdragon plants, CR. We have white ones, CW. Okay. And so when we mate these 2, okay, we're going to do our Punnett square exactly the same way as we normally would. Okay. So here we have CR-CW. And so these are all heterozygotes. They have one copy of each allele. Okay? So in incomplete dominance, the heterozygotes are going to have a blend of the 2 parent alleles. Excuse me. So in this case, we have CR-CW. So this would be red and white. Red and white is going to make pink. So these snapdragon plants would end up being pink. Okay. And so all of your offspring in this case would end up showing up as pink offspring. Okay. Now, if we were to take the F1 generation, okay, all of our pink offspring here, and we allow these two or two of these guys to mate with one another, okay, two pink offspring, okay, then we're going to see that the red and the white begin to reappear in the next generation. Okay, so here we have some pink parents, the CR2W, CR2W, okay, and we allow them to mate with one another. Okay, and so when they mate, okay, you again do your Punnett square just like normal. Okay, here we have CR, and so these would come up as red. Here we would have CR-CW, so we'd come up as pink over here again. Okay. Again, CR-CW, so we'd come up as pink over here. And then we have CW-CW, and so we end up with white. Okay. And so here we see that the red and the white plants, or red and white flowers, are able to appear again in the next generation, okay, along with the pink flowers. And so whenever you're crossing heterozygotes, whether you're looking at simple dominance or incomplete dominance or codominance, we're always going to see a ratio of 1 to 2 to 1, or a genotypic and phenotypic ratio, meaning that you're going to have one red homozygote, in this case, two pink heterozygotes, and then one white homozygote. Okay. One red, two pink, and then one white. Okay. So we see a 1 to 2 to 1 genotypic and phenotypic ratio here, whenever we're crossing heterozygotes. Okay. But again, here we're having a blend whenever we have heterozygotes in our offspring. Okay. All right, so here's an example. Okay, so here we have in humans, hair texture exhibits incomplete dominance. Okay, the gene for curly hair, right? This probably should say allele, the gene for curly hair, HC, is incompletely dominant to the gene for straight hair, HS. Okay. So individuals that are heterozygous have wavy hair. So what would their genotype be? Okay. If we have wavy hair individuals, what would their genotype be? Would it be HCHC, HCHS, or HSHS? So Brianna says B, and Brianna would be correct. Yeah, so if you're looking at a heterozygous individual, and Kimberly too, cool. So if you're looking at a heterozygous individual, they're going to have one copy of each allele, okay, the HC and the HS, okay, and that would give them wavy hair. Okay, so here's an example. So same thing. So we have Ned and Catelyn Stark both have wavy hair and have babies. So let's try to do their Punnett square from here. Okay. So again, we'll kind of draw out our Punnett square. Okay. And so boom, boom. Here we have our Punnett square there. Okay. And so if Ned and Catelyn Stark both have wavy hair, what would their genotypes be? Anyone know? HCHS, right. So Ned would be HC. Okay. HS. Okay, so I'll just put Ned over here. Okay. And then Catelyn Stark would be, I'll do that in a different color. She would also have wavy hair, so she'd be HC over here, and then HS over here. Oops. Okay. And then again, you would just do your Punnett square just like you normally would. Okay. So in this first box, what would we come up with? We come up with HC, right. So we have HC, HC. Perfect. Okay, what about the second box over here? HCHS. Beautiful. Okay. This third box over here. HCHS. Beautiful. And this last box on this side would be what? HSHS. Perfect. HS, HS. Perfect. Okay. So now if we want to interpret our Punnett square, let me go ahead and do this over here. So if we want to interpret our Punnett square, let's look at the genotypes first. Okay. So what is the likelihood that we're going to end up with a homozygous individual, just homozygous as a whole? %, right. So we're gonna have 50% homozygous offspring, or 50% chance of having an offspring that is homozygous. Again, because we're having HC over here and an HS over here. So we have 2 out of our 4 boxes being homozygous. Okay. And then what's the chance of us having offspring that are, heterozygous? 50%, right. So 50% chance of heterozygous offspring. Now, if we want to look at this, if we want to look at this a little bit more deeper, let's take a look at the phenotypes that we can see. Okay, so what's the chances of having an offspring that has curly hair? So it's not going to be 75%. 25%. So Brianna got this one right. So it's going to be 25% because you're going to be looking for the HCs. Okay. Remember, HC is going to give us curly hair, right? That's going to be a HC. Okay. What about the likelihood of having, I'll put straight hair? %, right. 25%, because again, you're looking for both HSs. So HS, that would give us straight hair. Okay. And then what's the likelihood of having an offspring that ends up having wavy hair? 50%, right. So 50%, and that's going to be where we look at the HCHSs. Cool. So remember, in people who have, or in individuals who have curly hair in this case, they're going to have 2 copies of the HC allele. Okay. If they need straight hair, okay, they're going to have 2 copies of the HS allele. Okay. And then because it's incomplete dominance, okay, where you're going to get a blend, okay, to have wavy hair, you're going to have a combination of both the HC and the HS allele. Okay. Okay. This is making sense so far. Cool. Okay. Any questions on incomplete dominance? Okay, cool. All right, so we'll go ahead and jump to the next one here. Oops. Okay, so this next one is called codominance. Okay, so codominance is kind of similar to incomplete dominance, but instead of having a blend of the 2 alleles show up, both individual alleles show up on the offspring. Okay, so this is where you have both alleles getting expressed at the exact same time. Okay, so instead of having, for example, well, I'll give an example in a second here. So an example would be blood types. Okay, so if you're like type A, type B, or type O blood, or type AB blood, we're going to see some codominance here. So here's an example. Okay. So here, in order to have type A blood, so your blood type is going to be also determined by genetics as well. Okay. So if you're a homozygote who has type A blood, your alleles or your genotype is going to be IA. Okay, 2 copies of the IA allele. Okay. If you're a homozygote who has the type B blood, okay, then your genotype is going to be IB alleles. Okay, your genotype. Okay. Now here's where the codominance actually comes into play. If you're a heterozygote, then you're going to have both the type IA allele as well as the IB allele. Okay. So both the A and the B proteins on your red blood cells are going to show up. Okay. So people who are just type A will have just the A protein. People who have type B will have just the B protein. But in people who are type AB, they will have both the A and the B proteins on their red blood cells, and that's going to contribute to them having type AB blood. Okay, so this is where we actually see codominance actually taking place. Okay, where you have the A allele and the B allele both appearing. You're not getting a blend, okay, but you're having both of them show up. Okay, and so that's how we end up with type AB blood. Okay, specifically by codominance. Okay, on a kind of a separate note, for people who are type O, okay, type O blood is going to actually be like directly recessive to the IA and IB alleles. So you can also have type A blood if you have the IA and O allele, because the O would be recessive to the IA allele. Okay? And so you'd end up with type A. And same thing with type B. Cool. So that's what we would see here. So we would cross someone who had a type A blood, who's a homozygote for type A blood, and a homozygote for type B blood. Okay. They eventually have a baby, and you can see they're going to have IA, IBs. And so as a result, they would be producing both the A and the B proteins on their red blood cells. And as a result, that individual would be someone who has type AB blood. Okay. So again, if you were to take two individuals who have type AB blood, okay, again, you're going to take the IA, IB on one side and the IA, IB on the other side. Okay, they're both going to be heterozygotes. Okay, if you cause those heterozygotes to mate, okay, again, we're going to see that same 1:2:1 genotypic ratio. Okay, where we're going to see one person with type A blood who's a homozygote, two people with type B blood who are heterozygotes, and then one person with, or sorry, type AB blood, and one person with type B blood who's a homozygote as well. Okay. So again, whenever you're crossing heterozygotes, we're going to see that same 1:2:1 genotypic ratio where you have a homozygote to a heterozygote to a homozygote. Okay. All right. So here's an example here. Okay. So here we've got some chickens. Okay. So in some chickens, feather is controlled by codominance. So when a black chicken, which has the FB allele, mates with a white chicken with FW allele, all the offspring are covered in both black and white feathers. Okay. So we have a situation here where we have a farmer that's going to mate a black chicken with a black and white chicken. Okay. So we're going to do the Punnett square here. Okay. So let's go ahead and draw this out. Okay. So here we have our Punnett square. All right, cool. Okay, so we're going to take a look at each of these chickens. Okay, this first chicken is a black chicken. Is a black chicken. Okay, so if we have a black chicken, I'm going to give up. Okay, if we have a black chicken, what is its genotype going to be? It was completely black. It's going to be FB, right. So we're going to have FB, FB over here. Okay. And if we have a black and white chicken, okay, what would its genotype be? FB, FW, perfect, Michaela. Yeah. So this would be FB, FW. Perfect. And then we do our Punnett square just like we normally would. Okay. So in this first box, what would we have here? So we got FB, 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 right. So FB, FB. Okay. What about in the second box? What would we have? Right, FB, FB. Perfect, Jared. So FB, FB. Okay. And then box number 3, what would we get here? FB, FW, right? Or FW, FB, doesn't matter which way. Yeah. So FB, FW. Okay. And then box number 4. FB, FW, right. FB, FW. Okay. So let's go ahead and interpret our Punnett square. Okay, so we'll start with our homozygotes. Okay, so what is the likelihood or the probability of having an offspring that is a homozygote? Right, 50%. 50% because, again, we're looking at two of the same alleles, okay, and we see that in two of the four squares, okay, these two top guys right here. Okay, what about the chances of getting a heterozygote offspring? Right, 50%, again, perfect. Again, we have two copies of different alleles, okay? FB over here and FW over here, and we see that in two of the four squares as well, okay? All right, now if we want to interpret this further and look at the phenotypes, okay, what is the chances of us seeing an offspring that is completely black? So a completely black chicken. 50%, right. So it would be 50% because, again, we're looking at our two FB, FBs. Okay. What's the chances of having a black and white offspring? 50%, right? Because, again, we're looking at the FB, FWs down here. Okay. And then what's the chances of having a completely white offspring? 0, right, because we don't see any FW, FWs. Perfect. Awesome, cool. Looks like we're catching on. That's good. All right, any questions on codominance before we jump forward to the next one? Okay, so if not, we'll go ahead and jump forward here. And so this last one is a little more complicated. And so this is where we're looking at sex-linked traits. Okay, so sex-linked traits are going to be traits that are only found on the sex chromosomes. Okay, either the X chromosome or the Y chromosome. And so we can see some inheritance patterns take place on the sex chromosomes, and typically a lot of sex-linked traits are going to be on the X chromosome. Okay, so those genes are what we're going to refer to as X-linked genes because they're only found on the X chromosome and not the Y chromosome. Okay, so these do get a little bit complicated. So just to kind of recap here, when we are looking at females, okay, females are going to have two copies of the X chromosome, and males are going to have a copy of the X and the Y chromosome. Okay? So sex-linked traits were first discovered in Drosophila flies, which is just the common fruit fly, and they kind of exhibit the same kind of pattern in terms of their X chromosome, their sex chromosomes. So female fruit flies have two copies of the X chromosome, while male fruit flies will have an X and a Y chromosome. Okay? So when these were first being discovered and looking at sex-linked traits in these fruit flies, what they started looking at was the color of their eyes. Okay? And so the eye color, the gene for eye color was found on the X chromosome, okay, for these fruit flies. And so there's two variations. There was an XR, which was the more common trait, and so that would give the red eye color. Okay, and then there were some fruit flies that had white eye colors. Okay, so they had white eyes, and so their allele would be X little r. Okay, so X meaning that it's found on the X chromosome, and then the big R, little r tells you whether it's dominant or recessive. Okay, so X big R was the dominant red allele. Okay, red eye allele. Okay, X little r was the recessive white eye allele. Now, when we're looking at sex-linked traits, we can't classify males in the same way as we've done previously. Okay? The reason for that is because they only have one X chromosome. Okay? There's no way for them to have two copies of an allele on the X chromosome because they only have one X chromosome. There's not another X chromosome to have another allele on. Okay? So for that reason, we call males hemizygous, okay? Meaning that they're only going to have one X chromosome and one allele for that particular trait, because again, you only have one X chromosome. Okay? And so they're either going to be X big RY or X little ry. Okay? So again, X because it's on the X chromosome, R, either dominant or recessive, and then the Y because they're also going to be carrying the Y chromosome also. Okay. Females are going to be classified the same way as we normally have. Okay, so females can be homozygous, either homozygous dominant, meaning that there can be either X big R, or they can be homozygous recessive with X little r, or they can be classified as heterozygous, where they have X big R and X little r. Okay. So classify them exactly the same way as we normally have. Males, we can't do that because we're only dealing with one X chromosome. Okay, so when we're looking at sex-linked traits, we're going to see different inheritance patterns depending on where we find that recessive trait. Okay. And so if that recessive trait was expressed in the male or in the female, okay, that is going to alter the different types of variations that we can get in the offspring. Okay. And so we're going to do a few examples to kind of illustrate what I mean here. Okay. So we're going to take a look at a set of crosses here. Okay, so here we have a white-eyed male, okay, X little ry, okay, crossed with a red-eyed homozygous female, okay, where we have X big R. Okay. So again, we're going to do this the exact same way. Okay, so we're going to draw our Punnett square. Okay, and so I'll go ahead and just put, it doesn't matter where you put the male, but I'll just put the male on this side, on the top. And then I'll put the female on the other side. I'll just put F down here. Okay. All right, so let me kind of finish our Punnett square here. So here we have our boxes. Oops. Okay. So if we look at the male, okay, the male is going to be X big R Y, or sorry, X little R Y. Okay. So we're going to put the X little r right here, and then we're going to put the Y in the other box. Okay. And then when we look at the female, we have X big R, so we're going to put along this box X big R, and then in the other box X big R as well. Okay. Now, and then we do our Punnett square just like what we normally would have. Okay. So if we look at this first box, okay, this first box, what would we get? Right, so Michaela got this one right. So we'd have X big R and then X little r. Okay. In the second box, what would we have? So Michaela almost had it. So this one would just be X big R Y. Right, so Kimberly exactly right. So X big R Y because you're just dealing with just the Y chromosome in this box. Okay. What about this third box down here? What would we get? So Jasmine got this one, so X big R and then X little r. Okay. And then in this last box, what do we come up with? X big RY. Perfect. X big RY. Beautiful. Okay. And so if we were to take a look at our offspring, so there's a couple things we need to be mindful of. Okay, so number one, we want to take a look at the whether we're looking at homozygous individuals or hemizygous individuals or heterozygous individuals, okay, just like normal. Okay, we want to be able to determine the phenotype, but we also need to keep in mind the sex of the offspring as well. Okay, so which of these four boxes are going to be classified as females? Is it going to be the two on the left, the two on the right, or sorry, the two on the left, two on the right, two on top, or two on bottom? Which ones are females? Right, the two on the left. Right, so these would be the females because we have are X chromosomes, or 2 X chromosomes. Okay. And then the 2 on the right, I'll just kind of do this in blue, or let me do this in a different color. We'll do this in red. This is where we would find our males, okay, because we have 1 X chromosome and then a Y chromosome. Okay, so looking at our Punnett square here, let's kind of look at our phenotypes as a whole. How many of our phenotypes, or sorry, how many of our offspring, okay, would we expect to have red eyes? What percentage? Yeah, so Hasmin and Brianna got this one exactly right. So 100% of the offspring will have, I'll just put 100% of offspring will have red eyes. Okay, and that's because we see all these guys are going to have the X big R allele. Okay, so they're going to have the dominant I allele. Cool. Okay, so we'll kind of leave it at that, and then as we kind of go through some other examples, we're going to get a little bit more specific here. Okay, so this is kind of what we would see here. So I'll go ahead and kind of clear this out. Okay, so this is what we would see, right? We would see all of our offspring having the X big R allele, so they're all going to be red-eyed offspring. Okay, so all offspring are going to have red eyes regardless of their sex. Okay, regardless of whether they're female or male. All right, let's change up the scenario a little bit. So now let's say we're looking at a red-eyed male, okay, and we're going to cross him with a white-eyed homozygous female. Okay. So again, let's go ahead and do our Punnett square here. And if we do our Punnett square, here's our Punnett square. Okay. So we have a red-eyed male, which is going to be X big R Y. X big R Y. Okay. And then the white-eyed homozygous female, so we're going to put her along the side. So she's going to be X little r, X little r. Okay. So now if we go through our Punnett square, this box we'd end up with X big R, X little r. Okay. The box on top right, we'd end up with X little r, Y. Okay. The third box would end up with the same thing, X big R, X little r. And the last one, we'd end up with X little r, Y. Cool. Okay, so now based off of our Punnett square, we'll kind of interpret this a little bit here as well. Okay, so let's start by taking a look at the offspring as a whole. Okay, so out of our offspring as a whole, how many of our offspring will have red eyes? Right, we're gonna have 50% because we see these two on the left are going to have the dominant allele. Okay, so they're going to have the red eyes. Okay, how many are going to have white eyes? How many offspring are going to have white eyes? 50%, right. 50% are going to have white eyes as well. Okay, so here both these guys are going to contain the recessive allele, okay, and nothing else. Okay. Now we're going to take this a step further. If we're looking at just the female offspring, what's the likelihood that the female offspring are going to have red eyes? What do we think? So I'm seeing 50. So I'm doing 50 as well. So this 50 isn't quite right. And so this is where things get a little bit tricky. You kind of got to think about this a little bit in more detail here. Okay. So when we're looking at our Punnett square, which are going to be classified as our females? Is it going to be the top, the two left ones or the two right ones? The two on the left, right. So I'll kind of put these guys in pink. Oops. Let me undo. Oops. Right. So yeah, so these would be our females over here. Okay. So these are our females. So based off of this, okay, if these are our females here, how many of our females are going to have red eyes? What percentage? We had two potential ones. So what do we think? Right, it'd be 100%. Right, so you have two options here, okay, and both of these offspring here contain the red-eyed allele, okay, the dominant red-eyed allele. So the females with red eyes, whoops. I don't know what I'm doing here. I don't know what I did. Oh, I screwed up. Okay, we'll just write this again. So females with red eyes. Females with red eyes would be 100%, because again, you're taking a look at these guys over here, just these two. Okay. And now if we're looking at the individuals that have blue eyes, or sorry, that have white eyes. All right, actually, let me backtrack. And so as a result, you'd end up with no offspring that have, no females that have white eyes. Okay? So all of your females are going to have red eyes, and all of your, and none of them are going to have white eyes. Okay? But if we look at the males now, okay, so we look at our males. Okay, okay, what's the likelihood, or what's the probability that our offspring are going to have red eyes? Okay. We'll say males with red eyes is going to be what percentage? 0%, right? And so the males with white eyes would account for how many? What percent? %, right, exactly. So this is where sex-linked traits start to become a little bit more complicated, because not only are you looking for the offspring as a whole, but you now need to take into consideration, okay, if I have a female, what's the chance that they're going to end up with this trait? If I have a male, what's the chance that they're going to end up with this trait? Okay, and so on and so forth. Okay. So this is where things get a little bit more muddy, and yet there's kind of an extra step to your analysis of your Punnett square. Okay. Actually, I'm going to go ahead and kind of clear this so we can kind of look at this in some more detail here. And so if we do our Punnett square, okay, this is kind of what we end up with. And so all of the offspring, or sorry, they're going to be red and white offspring. All the females that you end up producing are going to have red eyes, and all the males that we produce in this case would have white eyes. Okay. Now, if we do a different scenario, let's say we have a white-eyed male and we cross them with a red-eyed heterozygous female. Okay. Do we want to go through this, or kind of go through it step by step again, or do you want to just kind of flow through? Okay, we'll go through it. Okay, so if we go through this guy here, so this particular scenario, okay, so we'll go ahead and do another Punnett square. Okay, here we have a white-eyed male. Okay, so if we have a white-eyed male, we're going to have an X big R and a Y. Okay. So X little r and Y. Okay, because white is recessive. Okay. And then for the female, X big R, X little r. And X little r. Okay. Okay, good. I'm glad these examples are helping out. Cool. Okay. Now, in this first box, what would we end up getting? Right, we'd have X big R, X little r. Okay. In box number 2, what would we get here? X big RY, perfect. Okay. The third box, now we have X little r, cool. And then in this last box, X little ry. Okay. Good. All right. So again, let's go ahead and take our offspring, and we're going to look at them as a whole. Okay. So if we look at all of our potential offspring, what's the likelihood that we can have offspring with red eyes? Right, 50%, because we're going to see the dominant allele in these top two boxes over here. Okay, and so what's the chance of having offspring with white eyes? Again, 50%, right? So we're going to have ones that have the the recessive allele over here, and then just the recessive allele over here as well, 50%. Okay. Now, if we look at females, okay, what's the likelihood that our females will have red eyes? Right, 50%. Again, just because you're looking at just the females, okay? So you're gonna be looking at just these two boxes. Only this top box has red eyes. So that's 1 out of 2. That's gonna give us 50%. So females with red eyes. Okay. What about females with white eyes? What's the chances? Right, again, 50%. Perfect. Okay. Now, if we're looking at the males, okay, looking at males with red eyes, what's the chances we're going to end up with a male with red eyes? Right, 50%, because again, you're going to look at just these two male boxes. Okay, 1 out of these 2, that's going to count for 50%. So you have a 50% chance of having the red eyes, okay, if you're a male, okay, if you're a male offspring. Okay, and then what about males with white eyes? 50%, right? Exactly. So here we see as a whole, there's a 50% chance of having an offspring with red eyes and 50% chance of having an offspring with white eyes. Okay. If you're looking at just females, there's a 50-50 chance of a female having red eyes versus white eyes, and the same trend for males. Okay. And so again, we're starting to see different scenarios and different outcomes depending on where that initial recessive allele was found, whether it was found in mom or dad or both. Okay. Cool. So I'll go ahead and clear this so we can kind of see, so we can kind of see what's going on here. So same thing. So half of the females, half the males that we produce and half of the females will have red eyes and the other half will have white eyes. Okay. Okay, so I have one more example here. So we'll go ahead and do this last one together. And so this would be if we're looking at a red-eyed male crossed with a red-eyed heterozygous female. Okay. So again, if we do our Punnett square, so we have X big R Y for the dad here, and then we have X big R X little r, okay, for the mom. Okay. And so, okay, and this first box, what would we come up with? Right, X big R, X big R. Okay, what about this top right? X big RY, perfect. Okay, and then this bottom left, X big R, X little r. And then bottom right, X little RY, perfect. Perfect. X little R, X little, yeah, X little RY. All right, so now again, if we want to take a look at our offspring, okay, looking at our offspring as a whole, what would we, or what's the chances of having offspring with red eyes? Right, 75%, right? Because we're going to see 3 out of our 4 boxes are going to have the dominant allele. Okay. Perfect. So 75% chance of having an offspring with red eyes. Okay. What's the chance of having an offspring with white eyes? Right, 25%. Perfect. Okay. Now, if we're looking at females, okay, if we, let's say we have a female offspring, let's say we have a female with red eyes, what's the chance of having a female with red eyes? 100%, right? Because both of these two, okay, are going to have the dominant red eye allele. Beautiful. Okay. And so as a result, you end up with how many females would end up with white eyes? 0. Right. Perfect. Cool. Now, if we're looking at males, okay, what's the likelihood of having a male with red? Oh, I can't cut today. A male with red eyes. Right, 50. Right, because you have 1 out of your 2 boxes with the dominant allele. So 50% there. And then any males with white eyes, what's the likelihood there? 50%. Right. So here we can see that even though we had parents that both carried the dominant trait, okay, or both were expressing the dominant trait, we were still able to have an offspring that showed the recessive trait. Okay, we had a red-eyed parent over here and a red-eyed parent on this side, okay, but we're still able to produce an offspring that had white eyes. Okay. So again, depending on the X-linked trait and where that recessive trait shows up, that can determine what the offspring or what ratios the offspring can show up in. Okay. So I'll go ahead and kind of clear all this here. Okay. All right, how are we feeling with this so far? Yes, Punnett squares are fun. They're exactly like a puzzle. I've always loved Punnett squares. And, they're really cool. They're really cool, and it's a little puzzle. You have to have a little bit of fun with it too. It's a little mystery. Cool. Alrighty. So that is that. Okay. So again, when you're going through the Punnett squares, you got to be able to determine how many are going to be heterozygous, how many are going to be homozygous, how many are going to be dominant, recessive, and then if you're looking at sex-linked traits, also be able to do that for each sex, whether you're looking at males or females. Okay. So an example of color blindness that happens in humans, or sorry, an example of sex-linked traits that happens in humans is color blindness. Okay, so color blindness is a condition that is on the, that is on the X chromosome, and it's an X-linked recessive condition. Okay, so there are scenarios where a mother who is not colorblind, okay, she's still going to be classified as a carrier for it. Okay, so if she has just one X little b allele, one recessive allele for color blindness, she is considered a carrier. Okay, so she's a carrier because she herself does not express color blindness because she has the dominant color site allele over here. Okay, but she still carries the color blindness allele. Okay, this recessive one. Okay, and so as a result, she is still able to have an offspring with someone who has, or let me try to say that one more time. She is still able to have an offspring who is colorblind, even if the baby daddy isn't colorblind himself. Okay, so this is what we'd see if we were to take a Punnett square with a man who has normal color vision and a woman who is a carrier for color blindness. Okay, so here we would see X big B, X big B, X big B Y, X big B, X little b, and then here we would have X little b y. And so this would be the offspring that ends up becoming colorblind. Okay. Okay. So that being said, females are capable of inheriting the color blindness allele if they're able to inherit both of those color blindness alleles from their parents. Okay. Now, if the father is normal, okay, if the father has the color blindness allele, he can still have the dominant. Let me backtrack. So females, I'm getting all over the place. So females are capable of inheriting the color blindness allele, okay, from either of their parents. Okay. However, if the father is normal, okay, he's gonna have the normal allele, and it's gonna pass that on to the mom, and then she'll be able to mask the color blindness allele. Okay. So this is typically what's going to be happening in order to produce a woman who's a carrier of the color blindness allele. Okay. So that being said, and we'll talk about this more in detail in class after we come back from spring break. Okay, but males can be born with color blindness, as we see here, even if both parents have normal color vision. Okay, and that's going to be due to just inheriting the Y chromosome from their dad. Okay. So that being said, X-linked disorders, okay, which are usually going to be recessive, okay, are more common in males than in females. Okay, so color blindness, for example, is going to be more common in men than females. Does anyone have an idea why? Okay, kind of looking at our Punnett square, why would males be more susceptible to X-linked disorders like color blindness than females? Right. So Jared got it. So there's only one X chromosome. Okay. So in males, we have one X chromosome and one Y chromosome. Okay. So whatever X chromosome we get, that's all we get. Okay. That's all. That's pretty much it. Whatever we get is what we get, and that's it. And there's no backup. Whereas in females, if they happen to inherit, okay, a recessive allele, okay, they have the ability of having another X chromosome because they're inheriting one from mom and one from dad. That kind of acts as a backup. So if they're able to inherit just one normal allele, they're going to end up having the normal genotype and the normal phenotype. Okay. In males, we're not so lucky. Okay. Because we only have one chromosome, we don't have a backup, okay, like the females do. Okay. Whatever chromosome we get is what we get. So if the chromosome that we inherit is a healthy allele, good, we're hunky-dory, we're fine, everything's all good. Okay. But if we inherit a diseased allele, like color blindness, there's no way for us to come back from that. Okay, we have no backup. That's it. That's all we get. And again, that's just because we have one X chromosome and then the other one is our Y chromosome. So we're not so lucky. Girls, you guys have it good. Okay, this all kind of makes sense here. Any questions on Punnett squares and genetics and anything like that? Okay. If there's no questions here, sorry, this kind of went a bit long, but this is one that kind of takes a little bit of practice to go through. If there's no questions, I'm going to go ahead and show you what you're going to be doing for lab. So this is the lab 9 assignment. And so this is going to be due by 11:59 today. So is everyone able to see the lab assignment? Yeah, okay, cool. All right, so what we're going to be doing here is we're going to be doing just activity 1, 2, and 3. Okay, and then I'll talk about the last part in a second here. Okay, so this first part is going to be due today. And so in activity 1, you're going to be working through this table. Okay, and so there's going to be looking at you're going to be looking at PTC tasting. Okay, so to kind of give you a little bit of background here, what we would be doing in lab if we were together is I'd be giving you guys some PTC paper. And so the PTC paper is able to be either tasted or not tasted, depending on your genotype. Okay, so people who have the PTC tasting allele, okay, that's going to be the dominant allele. If you can taste the PTC paper, that's the dominant allele. Okay, if you can't, that is the recessive allele. Okay, so what you're going to be doing is you're going to be writing down the 3 possible genotypes for being able to taste PTC. Okay, so you're going to list what those genotypes are, okay? And then depending on what those genotypes are, you're going to determine what their phenotypes are, either tasting or not being able to taste it, okay? And so that's going to be for activity 1. Okay? You don't need to worry about circling it. I know it says circle your phenotype and your possible genotypes. You're not going to worry about that because we're not able to do this in lab today. So don't worry about circling or underlining or anything like that. Okay. And then you're going to answer the questions on number 2. Okay. So question 2, you're going to need to draw a Punnett square for that to be able to answer 2A and then B and C for that. Okay. And so you're going to write those answers in right over here. Okay. Okay. Any questions on activity 1? No. Okay. All right. For activity 2. Okay. Activity 2, we're going to be looking at color blindness. And if you remember, color blindness is a X-linked disorder, X-linked trait. Okay. So what you're going to be doing here is you're going to be filling in this table. Okay. So here we have the possible phenotypes, and you're going to list what their possible genotypes could be. Okay. So if they're female with color vision and they're homozygous, what is their genotype going to be? Okay. If they have a female with color vision with heterozygous, okay, what's their genotype going to be, and so on and so forth. Okay. So we're going to list what the genotypes are for all of these possible phenotypes. Okay. And then again, you're going to answer the questions for number 2 and number 3 in activity 2. Activity 3, we're going to be looking at blood types. Okay. So for activity 3, you're just going to be filling in this table. Okay. And this table is listed here. And so you're going to complete the table. So here we have some genotypes. Here we have whether it's going to be heterozygous and homozygous. And here we have the possible phenotypes. You're going to fill all this in. And remember that blood types are considered codominant. Okay. So this is going to be a codominant type of situation here. So list the genotypes, fill in the heterozygosity or homozygosity, and then whether it's what the phenotype would be, or the blood type in this case. Okay. So you're going to complete this, and then you're going to turn this that in by 11:59 tonight, okay, on Canvas, and that should already be up and ready. Okay. Now there's going to be some genetics practice problems that will also be due, but this is going to be due on the Monday after spring break. Okay. So this is going to be due on April 13th. And so if you go to page 73 and 74, okay, which are in your lab manual, okay, you're going to see a bunch of these Punnett square problems. Okay. Actually, I'm going to kind of get out of this real quick so you guys can see this a little bit better. So I'm going to go and stop share. Okay. So you're going to see all of these Punnett square problems. Okay. Make sure that you complete all of these. It's front and back, so page 73 and page 74. Okay. Do your best with it, and you're going to turn that in onto Canvas on the Monday after spring break, so April 13th. So that will be due. Do the best you can, because again, we're not going to be able to get to this particular chapter until after spring break. So like I said, just do the best you can with it, but just make sure you turn it in. Okay. Okay. Any questions on what's going to be happening for lab today? So you're going to skip activity 4 and activity 5 in the lab manual. Okay, we're just doing activities 1 through 3. Any questions on what we're doing for today's lab?